Welcome, biology students, to Chapter 2. This will be chemistry, where chemistry comes to life. Again, this is going to be a summary of a lecture that I would normally give in person. So because this is an online class, again, I would really encourage you to work through the MyLab and Mastering the Dynamics study modules for this chapter. Also, make sure you're doing your mastering assignments for each chapter worth 5 points. And you can, if you need extra practice, there's homework problems associated with each chapter. So I will only lecture for about maybe 15-20 minutes on this chapter. But again, please work through those dynamic study modules to get anything that we might have missed or have gone over a little more quickly. So chemistry has a special place in my heart because I majored in chemistry in college. But I'll try to keep this as simple as I can for you guys, and we'll try to keep it moving. First of all, everything that takes up space and has mass is called matter, and all matter is made of atoms, which are units of matter that cannot be broken down into anything simpler by ordinary chemical means. This is a look at an atom, and as we look at the atom, the protons and neutrons are little pieces that make up the nucleus of the atom. A proton is positively charged and a neutron is negatively charged. A neutron has no charge. The electrons have a negative charge. And electrons orbit around the nucleus of an atom, much like planets orbit around the sun in our solar system. And the electrons travel in shells or circles around the nucleus. And in this first innermost shell, we only have 2 electrons traveling around the nucleus. So we have 2 protons, 2 neutrons, and 2 electrons. And this refers to helium, which is an element represented by 2 on the periodic table because it has 2 protons, 2 neutrons, and 2 electrons. So this will be a neutrally charged atom because there's 2 positive charges in the protons and 2 positive charges in the electron, 2 negative charges in the electrons. So this is a neutral atom. So this is another look at helium, showing there's 2 protons in the nucleus, 2 neutrons in the nucleus, and 2 electrons in that first electron cell. If we look at oxygen, actually has 8 electrons and 8 protons and 8 neutrons. And in its nucleus, it has 8 protons and 8 neutrons in the nucleus, but the 8 electrons are split up between cells. When we look at the electron shells, where the electrons orbit around a nucleus, we can always have 2 electrons in that first shell because there's only space for enough 2 electrons. So the other 6 electrons, because I told you there were 6 electrons in oxygen, the other 6 travel in what we call the second electron shell that orbits the nucleus. So we have these elements that are made of different electron shells that are orbiting the nucleus, but only 2 electrons can be in that first shell, and the second shell can only hold 6. So if we get an element that's past oxygen that has even more electrons, we'll get more and more shells of electrons that are surrounding the nucleus. This is a review of those subatomic particles, where they're located, their charge, and about how much their mass is. An element is a form of matter that cannot be broken down into simpler substances. It's made of many atoms that are all the same. Examples are gold, iron, oxygen, and this is where we get into the periodic table. Each element has an atomic number and an atomic mass. The atomic number just refers to the number of protons in the nucleus. And for an element to be neutrally charged, meaning it doesn't have a charge, the number of protons always has to equal the number of electrons, because protons will be positive and electrons will be negative. Then the atomic mass is the number of protons plus the number of neutrons within the nucleus, because remember from that chart before, protons and neutrons are the ones with the mass. Electrons have a very insignificant mass. So this is our periodic table, and the atomic number will always be right above the element's symbol. So this is hydrogen, has an atomic number of 1, meaning there's 1 proton, and it also has a mass of about 1. So we go through the periodic table and we can see how each element is associated with an atomic number right above it. And again, that atomic number refers to the number of protons. And so if we look at oxygen, here we have 8 protons, and the number below it refers to the atomic mass. There are other interesting things about the periodic table. They're put into groups based on their properties. Some are metals, some are transition metals, some are non-metals. But again, each element has what we call an atomic number, which refers to the number of protons, and an atomic mass, which refers to the number of protons and neutrons put together. And if all of these elements are neutrally charged, the number of protons will equal the number of electrons as well. Elements with the same number of protons but different numbers of neutrons are called isotopes. And an example is the carbon atom. All carbon atoms have 6 protons, but we get different isotopes of carbon atoms that include different numbers of neutrons. So C12, carbon 12, has 6 neutrons because 6 plus 6 will equal 12. Carbon 13 has 6 protons and 7 neutrons because 6 plus 7 equals 13. And carbon 14 has 8 neutrons because 6 protons plus 8 neutrons equals 14. So same number of protons but different number of neutrons are called isotopes. Radiation is energy moving through space, such as radio waves, light, and heat. And a radioisotope is an unstable radiation emitting isotope. It can be harmful, causing the death of cells, or beneficial as used in medicine. So we know radiation is used in cancer treatment to try to kill off cancer cells. This is ultraviolet radiation from the sun, can cause sunburn. I'm sure we've all gotten that living in Southern California. Other examples of important isotopes in medicine: radioactive iodine can be used for thyroid imaging to help diagnose any sort of metabolic disorder. And like we said, radiation can help to treat cancer, as cancer cells are more susceptible to damage. This is an image of a normal thyroid gland and an image of an enlarged thyroid gland due to the radioisotope imaging that we can see. This is a logo for irradiated food. So if you ever see this logo, this means the food has been susceptible to radiation. All right, 2 or more elements may combine to form a compound, and a compound is usually different from those of the individual elements. The atoms in a compound are held together by what we call chemical bonds. The element sodium is a solid metal, and sodium reacts explosively with water. The element chlorine is a yellow gas, but when elements sodium and chlorine form, they form table salt, sodium chloride, NaCl, a compound quite different from its elements. So compounds, when they are combined together in chemical bonds, can be something completely different than the elemental components. Covalent bonds form when 2 or more atoms share electrons, and molecules are chemical structures held together by covalent bonds. Compounds are molecules formed with 2 or more elements. Molecules are often described in a chemical formula that can contain symbols for all the elements included in them. So again, if we look at hydrogen, carbon, and oxygen, we can see the atomic number is 1, having to do with 1 proton. The atomic number is 6 protons. And oxygen, the shell closest to the nucleus, can hold up to 2 neutrons, and the next shell out can hold up to 8 electrons, but the shell here just has 6. So atoms with more than 10 electrons have additional shells. Because the inner shell can only hold 2 electrons, the second shell can hold up to 8, but since this is oxygen, there's only 6 in it, because 6 plus 2 will equal 8. Anything more than 10 in the second shell, we would add on another electron shell. This is a look at some bonds and how they will come together as a single covalent bond, as a double covalent bond, where more than 2 electrons are shared, and a triple covalent bond. So each time you see 2 electrons being shared between elements, that's 1 single covalent bond. If we get more than 2, so here's 4, we'll get a double covalent bond, and then a triple covalent bond will be 2 sets of 3 electrons being shared together. Ions are atoms or a group of atoms with a positive or negative electrical charge. And an ionic bond results from the mutual attraction of oppositely charged ions. So opposites will attract if there's a positive or negative electrical charge difference. Ionic bonds can be much weaker than covalent bonds because they don't actually share electrons to form that strong connection. They just are kind of mutually attracted to each other because they're oppositely charged. So this is a look at sodium chloride. It gives up one of its electrons, so sodium becomes a positively charged ion, and having received an electron, chlorine becomes a negatively charged ion. So the oppositely charged sodium and chloride ions then are attracted to each other. The positive and negative come together, forming sodium chloride or table salt. When electrons of a covalent bond are shared unequally, the bond is called polar, and the resulting molecules are called polar molecules, and water is an example of a polar molecule. A hydrogen bond then forms when the slightly positive hydrogen atoms of one molecule are attracted to the slightly negative oxygen atoms in another water molecule. Hydrogen bonds are the weakest of all bonds, as we can see here in water. It's formed when an oxygen covalently bonds and shares electrons with two hydrogen atoms. But because the electrons are unequally shared, oxygen carries a slight negative charge, and the hydrogen atoms carry a slight positive charge. So we get what we call a polar molecule, where one side of the molecule is more positive and one side is more negative. And it gives it this kind of bent appearance to water. This is showing how hydrogen atoms form one water molecule. They're attracted to the oxygen atoms of other water molecules because opposites attract. So the negative ends of water will be attracted to the positive ends of water and vice versa. And this relatively weak attraction shown by those dotted lines is called a hydrogen bond. Water has really unique properties. It has high heat capacity, high heat of vaporization, and it's superior as a dissolving agent, meaning it's the best liquid to dissolve anything in, and that it can be traced to its polarity. This is a great video on the unique properties of water that I'll let you guys watch on your own. Again, I've attached all PowerPoints in the module as well. Then we'll get to acids and bases and how they react differently to water. Acids will release hydrogen ions when placed in water, and bases will release hydroxide ions when added to water. pH measures the hydrogen ion concentration. So if we have an extremely low pH on the pH scale, the greater the acidity of that liquid is. If we have a higher pH on the pH scale, the more basic a solution. So here's a pH scale. Neutral is about 7, so anything less than 7 we call acidic on the pH scale, and everything greater than 7 we call basic. Some acidic things, your urine is slightly acidic, beer, battery acids. Human blood is just about neutral on the pH scale. Baking soda is basic, ammonia is basic, and oven cleaner is basic. Buffers prevent dramatic changes in pH, and these are incredibly important. Many body fluids have the buffering capacity to maintain a stable environment. So buffers will just prevent any dramatic changes in pH. Biological macromolecules are what we call the giant molecules of life. Long chains are called polymers, made of repeating units of monomers. So the word poly means many, and mono means one. When monomers are joined to form polymers, so when one comes together to form many, water will be removed, and the reaction is called a dehydration synthesis reaction. In the same way, when many polymers are broken apart, water is added to that, and the reaction is called hydrolysis because you're hydrating it with water. Here's showing how polymers are formed by dehydration synthesis. So we're removing water to connect two monomers together to create a polymer, which is 3 put together. We can break polymers down by hydrolysis, adding water, in which the addition of water disrupts the bonds between the two monomers. This is another great video showing the difference between a polymer and monomer that I'll let you guys watch. Carbohydrates are polymers made up of a monomer called a monosaccharide. Saccharide has to do with sugar. Carbohydrates are your body's fuel. They're composed of only carbon, hydrogen, and oxygen, and they're classified by size into monosaccharides, oligosaccharides, or polysaccharides. Monosaccharides are simple sugars, and glucose and fructose are main examples. Oligosaccharides are a few monosaccharides joined together. One specific type is a disaccharide. Di means two monosaccharides are put together, and sucrose and maltose are examples. So basically, all of these monosaccharides, oligosaccharides, are just chains of sugars put together. Here's a look at the straight chain formula for glucose. And when it comes together, a ring structure forms of glucose in which carbon atoms with the ring are designated with the letter C, and a ring structure of glucose in which the C for carbon atoms within the ring is omitted. So this is a typical structure in chemistry, how we would construct glucose. This is showing how one monosaccharide can join with another monosaccharide to create a disaccharide. So one glucose plus one fructose equals sucrose, a sucrose disaccharide. Polysaccharides, again, poly means many. These will be chains of monosaccharides that store energy, provide structure. In animals, the main polysaccharide is glycogen, where humans store all of the sugar, and glycogen is mainly located in your liver and muscle cells. In plants, starch is the main polysaccharide. So here is glycogen, and again, it's a polysaccharide, so we'll see several monosaccharides joined together, and glycogen is stored in the liver. When we need extra sugar, glycogen will be broken down. Cellulose is a structural polysaccharide found in plants. This is cellulose. I'm going to skip over this. Humans lack the enzyme necessary to digest cellulose. However, it's still important in the fiber in the human diet. Lipids then are water-insoluble molecules made of carbon, hydrogen, and oxygen. Insoluble means they cannot break apart in water. And you know that if you put oil, which is a lipid, in water, it'll float to the top. Lipids help to protect vital organs, form cell membranes, store long-term energy, and they can be subdivided into triglycerides, phospholipids, and steroids. Triglycerides are polymers made of 3 fatty acids. Examples are fats and oils, and they can be saturated or unsaturated. Here's a fatty acid bonds to glycerol through dehydration synthesis, where water is taken out. Here's an unsaturated fatty acid, and the presence of a double bond right here between carbon atoms indicates an unsaturated fatty acid. And saturated fatty acids do not have any double bonds. So we call them saturated because they can stack on top of each other more easily. And that's why you don't want to eat a lot of saturated fat, because saturated fat, these long chains of fatty acids, can stack on top of each other, and it's harder for them to be broken apart and digested. Whereas unsaturated fat, because they have this little kink or angle to them, they can't stack as easily on top of each other. So that's why you want to eat more unsaturated fat. Another great video about lipids, again, watch on your own. Phospholipid molecules have a glycerol head that is polar and a hydrophilic part that hates water and mixes with water environments inside and outside of the cell, or hydrophilic, it loves water, and a fatty acid tail that's nonpolar and hydrophobic. So it fears water, and these will point inward and help hold a membrane together. So this is a look at how phospholipids come together to form cell membranes. The non-polar hydrophobic or water-fearing tails come together to form what we call a lipid bilayer, which are 2 sets of phospholipids. The hydrophobic tails join together in the middle, and the water-loving polar hydrophilic heads are connected to water on the outside. And this forms many cell membranes in animal cells. Steroids are lipids that consist of 4 carbon rings attached to molecules. Estrogen and testosterone are examples of steroids in females and males, and as well as cholesterol, which can be a risk factor for heart disease when found in high enough levels in the blood. Here's cholesterol, steroid, and estrogen, and a testosterone. So you can see here, without knowing much chemistry, how they kind of have similar structures because they're all steroids. Proteins are made up of amino acids, and amino acids are the building blocks of proteins. They'll consist of a central carbon, an amino group, and a carboxyl group, in addition to a unique side chain. So this is an example of an amino acid: the central carbon, the hydrogen, the carboxyl group, the amino group, and the variable side group chain. Amino acids form proteins. They're linked by bonds called peptide bonds. Chains of only a few amino acids are called peptides, and chains of 10 or more amino acids are called polypeptides. So you can see here, through a dehydration synthesis, taking out a water, how a peptide bond will be formed between 2 amino acid groups. Proteins are made of polypeptide chains of at least 50 amino acids. Proteins are incredibly important in our bodies for providing structure, transport, and movement. And these are the 4 distinct levels of structure that affect their function: primary, secondary, tertiary, and quaternary. We can see here each amino acid is depicted here as a bead with a polypeptide chain. This is primary structure. Secondary structure, such as the alpha helix or the beta sheet, results from the bending and coiling of a chain of amino acids. And the tertiary structure, this might be a test question, hint, hint, is the 3-dimensional shape of proteins when the alpha helix and beta sheet proteins come together and form a 3-dimensional shape. That's tertiary structure. Some proteins have 2 or more polypeptide chains, and each chain forms a subunit. So quaternary structure results from the attractive forces between 2 or more subunits. But tertiary structure refers to the 3-dimensional shape. Another great video. Again, please watch on your own. Changes in the chemical environment of a protein can cause it to lose its structure, resulting in a loss of function. And this is called denaturization. And this can occur either through temperature or pH changes. Enzymes are proteins that speed up chemical reactions. Without enzymes, chemical reactions would occur too slowly. An enzyme can't make a reaction that can't take place. And its specificity is due to the unique shape of each enzyme's active site. An enzyme binds to substrates at the active site, forming this enzyme-substrate complex. And the substrate is converted to 2 or more products. So enzymes help speed up the rate of chemical reactions, but they're never used up in the reaction. So an enzyme has a little active site right here where a substrate will bind to form an enzyme-substrate complex. It'll help the reaction proceed more quickly, so we get products out at the end. Here's a decomposition reaction where it decomposes the substrate into 2 products. And a synthesis reaction involves an enzyme where it takes 2 substrates, a beginning product, puts them together, and forms an end product in a synthesis reaction. Nucleic acids, 2 examples that we'll focus on are DNA and RNA. Deoxyribonucleic acid and a ribonucleic acid. A nucleotide is made up of a 5-carbon sugar bonded to 1 of 5 nitrogen-containing bases and a phosphate group. Here is a nucleotide made up of a sugar, a phosphate group, and a nitrogen-containing base. And nucleotides will make up nucleic acids. Here is the differences between ribonucleic acid and deoxyribonucleic acid. DNA is what carries our genetic material. The sugar is a little different. It's missing 1 oxygen. The bases are a little different, and the number of strands are 1 in RNA and 2 in DNA. So this is a look at a strand and how they're connected to each other, and the different bases: cytosine, adenine, guanine, and uracil. And this is a look at RNA. DNA carries our genetic material, and it comes together when hydrogen bonds are formed between the bases to form this helical twisted pattern of DNA that carries all of our genetic material. The special nucleotide is ATP, adenosine triphosphate, and this molecule is very important. You'll hear about it a lot in this course. It stores energy in its phosphate to phosphate bonds. So all energy from the breakdown of molecules such as glucose must be channeled through ATP before the body can use it. And ATP is often described as the energy currency of the cells. So ATP is just how our cells do everything they need to. They use the energy currency of ATP. Here's adenosine triphosphate. It's made of an adenosine group and 3 phosphates put together. When this phosphate bond is broken, we get adenosine diphosphate, plus that phosphate group that was broken off, and energy. So these high-powered energy bonds are between these phosphate groups. And whenever a phosphate group is broken off, we release a lot of energy that your cells need to function. Nucleotide video, please watch this one. And after this, you should be able to describe, the characteristics of subatomic particles. So you should know the differences between these, explain the structure of an isotope. You should be able to differentiate between these type of bonds, listing the unique properties of water, understanding the pH scale and which values indicate an acid and a base. You should understand what dehydration synthesis looks like and hydrolysis looks like. And you should be able to understand the general structure of these macromolecules in a cell and describe how ATP is the energy currency of the cell. Thanks for your time, guys. We'll see you for the next lecture. Take care.